From Microbe TV, this is Immune, episode number 66, recorded on March 27, 2023. I'm Vincent Racaniello, and you're listening to the podcast on the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome. Great to be back.
We're not in Hawaii.
We are not in Hawaii anymore. Not in
So, on Twin this week, Ori was in Hawaii.
Oh, really?
Yes. He was there on vacation. It was pretty funny. It was all echoey and stuff. Also joining us from Cleveland, Ohio, Steph Langle.
Hi. Hello, everyone. Good to be back.
Is Cleveland good? Everything cool in Cleveland?
Oh, it's cool. Yep. It's raining and 45 degrees. I just had a research associate start in my lab who moved here from Malaysia like 48 hours ago. And so she's having to adjust. It gets warmer here, though.
Big adjustment.
For her, it's quite the adjustment. Yes.
Cool.
But that's exciting.
Yes.
Neat.
Yes. She's great. Going to lead the B-cell core.
Yay.
Yay.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi. Great to see everybody. My app says it's raining, but my eyes tell a different story. It looks pretty sunny out there.
Nice.
I can't tell you what it is here. I forgot my phone in the other room, and the windows are boarded up. But when I came, it was very nice this morning. It's supposed to go up to 12C.
Nice.
All right. Today we have an interesting review article for you.
Yep.
It was actually an issue of immunological reviews with quite a few interesting review articles, which were published last year. And this one is by two people who have been on TWIV multiple times, actually. Immunological Memory to SARS-CoV-2 Infection and COVID-19 Vaccines by Alessandro Sette and Shane Crotty, both from the La Jolla Institute for Immunology, where it's always 72 and sunny.
Yeah, we don't want to hear their weather.
So I thought this would be fun to go through because if you're always wondering, what is it if I get two vaccines or three vaccines or infection and vaccines and vaccines and infection, and how's my immunology? And so we have quite a bit of data. In fact, what they say here, which I think they have a couple of gems here in this paper, which I'm going to point out. There are more data on human antigen-specific immune responses to SARS-CoV-2 than any other acute pathogen. And so they're basically like, this memory to SARS-CoV-2 is now a benchmark in human immunology.
Yeah.
Would you guys
absolutely.
agree that it's kind of establishing a lot of paradigms for immunology that we didn't know before?
Oh my gosh, yes. I mean, we are seeing so many more people being measured in real time with the most sort of up-to-date technologically advanced assays to look at these things. We have answers to questions for SARS-CoV-2 with memory that we don't have for many other pathogens, especially in humans.
Yeah.
Although to give credit, so if we're speaking about just acute infections, which seems to be the defining sentence, right? I think influenza had done a really good job of taking human samples and starting to define correlates of protection. So they have a very standard assay called the hemagglutinin inhibition assay, and that's a benchmark for vaccine studies. But I think what influenza studies didn't necessarily get these numbers of people, right? I mean, we just had this opportunity, and individuals accumulate memory over time, and you can't always get people before they're infected. You have to get them when they're like 6 months old, right? To do that. So this really allows for seeing what a serologically naive population looks like when they get acutely infected with a virus.
Yeah, I think you hit the nail on the head there. So many more people were willing to enroll in these studies, and the studies were much huger in nature, many more individuals. And just the fact that you could look at a naive population that only got a vaccine, or that got infected in the vaccine, or got the vaccine that infected. We could never ask those questions before in the population.
Right.
So it really enabled studies that were not possible before.
And of course, many immunology labs came and started working on the virus, right? That
Almost every
it had to be. It had to be. I mean, there are two motivations. One is people want to get answers, and That's what we do as scientists. But also, you always want to be funded, and it was a good way to be funded. And so you do two things at once.
Yeah.
We hope that the first predominates, of course.
Yeah, and I think credit to NIH is they allowed people to use the funding they currently had to do whatever studies were necessary.
Yeah, sure. They had
because if
right?
we had to apply for the funds and, pitch the projects, it would have taken prohibitively too long to do.
Yeah. It was also sort of nice because it meant that during some of the time when labs were not always up and running, one of the ways people stayed employed was by doing SARS-CoV-2 studies.
Yeah,
and during the first year, we could not be in our lab unless we were doing COVID-related stuff, right?
The other thing to appreciate, and they bring this up a couple times, is in terms of vaccine-induced immunity and an individual who may have been serologically negative, we relied a lot on the yellow fever virus vaccine to define some of these long-term, like how long do antibodies last, B memory cells. And so this is that's another thing that SARS-CoV-2 has brought.
And that's not a good metric because that's a very different infection, right? It's an infection with a viremic stage, which exposes many more lymph nodes to antigen, and it's delivered by a mosquito. So these viruses with viremic stages tend to have much longer memory than respiratory mucosal infections.
Right. And I just meant the vaccine because I agree with you.
Yeah, that's all we had.
But it is it was a different platform too for that.
I was going to say exactly that. Looking at a very different type of vaccine platform and being able to kind of think about what types of things are common across platforms or might be different between different platforms.
Yeah.
I mean, as you say, Steph, influenza was really the leader before, but the vaccines aren't so great. The COVID vaccines are much better than the influenza vaccines are.
It does seem that way, although maybe we would have to look at like what our goals are because you have circulating strains that are different that, I don't know, we could go into that, but you're right. I do broadly agree. I
think we've done pretty well against all the variants as they say in this paper,
Yes.
What is it? There's this, they use words that are amazing ability of the immune system to adapt to variants that it hasn't even seen yet. It's great.
Yeah, that was pretty cool. I like that. Yeah.
Yes. So I think what I meant to say is that the SARS-CoV-2 vaccines have been successful, and it's difficult. A lot of people say, oh, the flu vaccines are, barely they work if they're not matched. So I think those are two different outcomes, right? It's two different because influenza has multiple circulating strains. But
so does SARS-CoV-2, but I think, the flu vaccines.
But they're variants, right? They're not strains. We wouldn't say that.
They're variants, yes.
Or SARS-CoV-2 has multiple strains.
And so we have a lot more experience with influenza for sure.
Yeah.
And the flu vaccines are not as bad as everyone makes them out to be. As Daniel Griffin says, you may get flu after you're vaccinated, but you don't die most of the time. So they are working against severe disease. So
yeah. And then to give credit, before we dive into the paper, to the HIV field, I do feel like the technologies, the trainees that came out of those labs, the big HIV labs, they're able to switch and apply those technologies to learn about SARS-CoV-2. Shane Crotty was working on HIV vaccines, Michelle Nussenswag. So it's very set to, Alexander. So and I think that goes back to, where is the funding to help answer these questions, and then can we apply it to other things?
Yeah, it's important that they say this is the most we know about a particular acute infection.
Right. We don't want to anger the HIV people. Right. Something going on.
I think, I had to do something. I think that people who did work on HIV were happy to work on a virus where the vaccines work.
Yeah.
Right.
There's a lot of years of frustration with HIV.
It's a lot easier with SARS-CoV-2. Yeah, for sure.
So a little summary up front before we dive into specific parts. So we're going to talk about memory. So the article is immunological memory. That's what we're mainly going to focus on. You can be infected, which they call natural immunity. Okay. I don't know. Should we call it organic immunity? All right, we'll call it natural immunity. Then we have vaccination. And then if you have both, it's called hybrid immunity. And as they say, they each have their distinctive characteristics. But overall, multiple studies have shown that infection gives you about 80 to 95% protection against symptomatic infection for about 8 or more months for the original ancestral SARS-CoV-2 and through Delta.
Yep.
And significant but less protection against symptomatic disease caused by Omicron and its subvariants.
And the audience should probably assume that if we do not clarify, we are mostly going to be referencing symptomatic disease and severe disease. If we're talking about protection against infection, that's a very different metric, and we will state that. But most of these studies, and even when they do make these statements, sometimes they don't have the comparator or they don't say from severe disease, but that's usually the metric that we're talking about.
Right.
Yeah.
So the many people have gotten 2-dose mRNA vaccines. These give you high levels of protection, again, severe disease through Delta. But they say the high levels of immunity, they use the word wanes. Okay, so I'll use wane. I like contract, but it goes down over a period of months. And if you're talking about the 2-dose Chadox, the AstraZeneca Oxford vaccine, it's lower and it seems to go down faster than the immunity conferred by mRNA vaccines. With hybrid immunity, the antibody titers and the breadth of recognition of those antibodies are really high, higher than in previously infected people who get at least 1 dose, and in people who get at least 1 dose and who are previously infected. So vaccination plus infection also gives you very good immune responses. And we'll look into those in some detail in a bit.
I really like here that they actually specified the difference between vaccination and then subsequent infection, or infection and then subsequent vaccination, and give the references on those, because those are things that people often smush together for obvious reasons. And it's nice to be able to know exactly where to go to find the information on different situations.
They do seem to be qualitatively different based on the data.
Yeah.
So they say hybrid
It would be
immunity.
nice if we had 2 different names for hybrid immunity, whether you were vaccinated first or infected first.
Yeah.
Hybrid immunity results in more robust protection than either against disease than either infection immunity or vaccine induced. So but each of them, So we have infection-induced, vaccine-induced, and hybrid. They each have distinct characteristics of memory, and of course, that's what's important here. And they say, we know that high circulating neutralizing antibodies can give you protective immunity, but we don't see high titers necessarily, particularly after infection. And they say there's probably a lot of role for, of course, T cells, memory cells, B memory cells, and local tissue immunity and protection. So we're going to explore some of those.
And now you said waning and contraction, and I thought that was an interesting thing, because I don't know what you guys think, Steph and Brianne, but I think of contraction as a natural thing that's supposed to happen during an immune response. But the waning part is, we would hope it doesn't wane too much, that once you get the contraction, you'd stay there. And so do you think there's a difference, or would you call them the same thing? And do waning, we typically are saying like measurable things, and when we talk about contraction, we're talking about the immune response part, or would you define them differently?
I use them similarly. So I think of waning as just another way of thinking about contraction, mostly because I have a hard time determining what is correct or appropriate waning and what is inappropriate. In some cases, they're all just the immune response contracting. We can wish for more or less waning, but I'm not sure that we can quantitate what is sort of good and bad. And so I think that they're all the same as far as we know right now.
I have a little bit of a contrasting view, and this is based on personal experience of questions I get after talks, interactions with other immunologists. I'm very hesitant to use the word contracting, because the B memory cell response continues to increase over time, depending on the lasting of an antigen in a germinal center. So while I would refer to antibodies waning, but the B cells are not contracting after an infection or a vaccination. And I have this is my experience based on interactions I've had in the past couple of years, where I am very hesitant to use the word contracting because I don't know what's going on in the germinal center, and B memory cells do not contract until, they're going on for 6 more months. So that's my kind of flavor to it.
But would you say the overall number of antigen-specific B cells contracts?
Nuisance-wise paper showed that they kept going up after 6 months. So maybe it depends on the time post-infection or vaccination, and how long the antigen lasts. I guess if we would say over a lifetime, probably, or maybe over 2 years they contract. But if I'm looking at like 6 months out, the data is looking like in the germinal center, they were increasing. So that's why I get it. It's tricky.
Just memory or all B cells?
Memory.
And
that's where I think the
I think Brianne was asking
what
all B cells. Yeah.
Well, all B cells for sure wouldn't all be contributing. To like short-lived plasma cells are going to die off, right? So those are dying. They're not contracting. So I guess it just depends on what are we the nuance of that.
In immunological terms, in all situations, antibody levels in serum and mucosal
they do wane.
They do go down, right?
Yes.
Because you don't want to keep high levels. And so it's going to be different depending on the vaccine. It's going to be different depending on the agent, right?
Yeah.
And those differences are interesting to explore, like what makes it short versus long, right?
Yeah. If there's absolutely no contraction, that usually leads to autoimmune diseases like ALPS or something. So there has to be some contraction of some populations, while other populations, Steph mentions,
because you would have
contract.
too many antibodies in your blood if they didn't contract, right?
Yeah, and you'd have no more resources for the next set of cells to expand.
There'd be a caloric cost.
Because they talk about how you have this increase after mRNA vaccination or infection. And that usually is the contributions of antibodies from both short-lived plasma cells, which are usually not germinal center derived. They're not as affinity matured. Then you have these long-lived plasma cells. It's going to take time. And so the decrease is maybe those short-lived plasma cells are not contributing anymore, but then there's a plateau. And I think then this homeostasis of the long-lived plasma cells just secreting antibodies has now leveled off and is at some level for long-term. So, but yes, I do agree that we can't keep pumping out antibodies to SARS-CoV-2 at high levels forever. They do go down.
All right, CD8 T cell memory, first of all. So we know from other
T cell people are excited.
Viruses that these T cells are important for controlling and particularly clearing viral infections. And there have been studies which say, they're important for SARS-CoV-2. For example, one study, they cite early CD8 T cell responses are associated with milder disease. And there are other studies that show this correlation, but there's also one in non-human primates where they found that depletion of CD8 cells affects, in animals recovering from COVID, affects immunity against a rechallenge.
I always love when they can do that. And monkeys are probably the most relevant, but they're the most expensive. But you can see, CD8 T cells, you take them away. It's not great.
No.
I don't know if they're always the most relevant, but that's the closest, right? We did a paper on TWIV a few weeks ago showing that non-human primates, you infect them with the 1918 influenza virus, they barely get sick, right?
Yeah. And with COVID, and with SARS-CoV-2.
But especially in animals that where we can actually do things like deplete CD8 T cells, they're a particularly good model.
Yeah.
So you have to know your limitations, as somebody said in some movie sometime. All right, so let's talk in specifics. 70% of COVID cases, a month after infection, you can see SARS-CoV-2 specific T cells. Then that goes down to 50% by 8 months. So that's interesting. So half of people after 8 months don't have SARS-CoV-2 specific T cells. I presume they're talking about effectors here, right? Detectable,
They're just talking about detector CD8s.
Yeah. It's a lot of people. When millions of people are being infected, that's a lot of people that don't have it. It's an interesting, even at 1 month, 30% of people can't detect them. They probably have some, right? But you're just not detecting them.
Yeah.
But they're surveying the blood, right?
That's true.
So
and they make that point
it is,
later. Yeah.
You know, it is the major location in which we can sense them, you know, or detect them. But there should be still some in the lung.
Yeah, which are harder to assess, as they say later. Yeah.
Right.
Unless you're willing to give.
And we only are as good as our technologies, which are pretty good.
Right. Yeah. They also mentioned that, you know, it's possible that there could be responses to epitopes that were not detected or not really well tested here, or this could be sort of a limit of detection issue.
Yeah. So they give us some half-life numbers on SARS-CoV-2 specific memory CD8 T cells. In one study, it was between 125 and 190 days. That's the half-life. And in another study, it was 196 days. And they say that's pretty good considering they use different assays, right?
Mm-hmm.
And in comparison, for yellow fever vaccine, the half-life is 123 days. So, and they say, you know, the yellow fever vaccine is good. So these are good numbers to do. So they say infection gives long-lasting SARS-CoV-2 specific memory CD8 cells, and they cite the fact that we know memory CD8 cells to SARS-CoV, the original, have been detected 17 years after infection.
Right.
That's a really good one because there have been no more infections since that original SARS-1, right? So you're not getting any boosting, whereas it's harder to know with SARS-CoV-2, as Danielle Weisskopf told us, right?
Yeah.
Because people keep getting infected, so it's hard to know.
Do you know from that interview with her or from other studies, do comical coronaviruses tickle the T cells?
They do.
Against SARS-CoV, the original. So you know.
That's harder to know, right? Because you don't have
those samples
numbers.
are harder.
The numbers are harder. But for the common cold, we're going to come to it.
No, I know for SARS-CoV-2. Yeah, we'll talk about it. But I just was curious about SARS-CoV-1 because that would be interesting.
It's hard. I think it's hard because even for SARS-CoV-2, you know, we're talking about 5 or 6 people that they've been able to look at. So there's going to be even fewer for SARS-1. So I think it'd be really hard.
Hard to find. Yeah.
And no one's going to do the challenge. So even though we do have SARS-CoV-1 in a lab somewhere, in several labs, Anyway, these
BSL-4, I'm assuming, right? Would that be a BSL-4?
It's actually it's a BSL-3.
Okay. I just didn't know because it was technically eradicated if that mattered, you know, because we don't, it's not circulating anymore.
No, it is a BSL-3, but it is a select agent.
Okay. That's the difference.
That makes it, you just can't work with it in any BSL-3. You have to have FBI clearance, and that's hard.
Got it. Okay.
Right?
Yes.
Select agent, yeah. Anyway, they talk a little bit about these kinds of CD8 cells. They mainly produce interferon gamma and granzyme B, a little bit of TNF, IL-2. And 8 months after infection, then we have effector memory, the TEMs, and another kind, TEMRA, predominating, and fewer central memory phenotype T cells. By the way, there are nice pictures in this review. I'm sure it's open access, so it would be cool to look at it. They talk a little bit about exhaustion, and they say that in their view of the literature, exhaustion doesn't seem to play a role here.
I'm glad that they clarified that. I think that a less nuanced view on PD-1, which is a marker that can be used as an exhaustion marker, particularly in cancer studies, just that alone is not definitive. That's not sufficient to tell you if a T cell is exhausted because it can be expressed in non-exhaustive conditions.
Yeah, so they do a nice job of sort of talking about expression of PD-1, but also whether they can measure other functions of those T cells.
We just had Arlene Sharp here, who originally found these PD-1 and characterized all the PD-1, PD-L1, PD-2. And she was very clear about, you know, we all forget that PD-1 is a molecule that's very rapidly upregulated on activated T cells. So, you know, calling it an exhaustion marker isn't really right. And a lot of her studies have really narrowed down that it doesn't work exactly the way we thought it worked. And there's more to learn about the different exhaustion markers. There's more than just PD-1 and PD-L1.
Mm-hmm.
So it was pretty cool.
Yeah. So they reiterate that what we already said, 30% of people of COVID cases don't make, well, we don't detect SARS-CoV-2 specific memory CD8 responses, even though we look at the entire, proteome of the virus, where we got all the peptides in there. So why that is, we don't know.
Yeah, that is odd. I mean, I don't have a deep knowledge of CD8 T cell literature, but do we know why that is? Is that common amongst other acute viral infections? I
don't know.
It seems like this cell matters, right? I mean, it can kill infected cells. It seems to contribute in animal studies and epidemiological studies, but that would be a determining factor for, let's say, your antibodies have waned. Let's say you don't have a robust mucosal immune response to help bring down those titers. So you got to rely on the CD8 T cells, but maybe you're older, they are defective when you age. So it's just interesting to me.
Yeah, I'm not sure how many other viruses have had all of the peptides across their open reading frame tested in this way. And so when I think about particular studies, I think of studies that say this percent of people with this MHC class 1 type will have the dominant or subdominant epitopes. But looking at a population level and looking at all of the peptides across the entire virus is not something I think that we've done very frequently.
That's interesting. Yeah, I agree. I'll look up and see if there are any influenza T cell people or HIV T cell people to see if, HIV is different. So maybe I should stick with flu people, right? Because it's
sure.
a better representation. Okay.
Do we know, what about the virus kind of interfering with the T cell response? Could it be that in some of these individuals that we're not able to detect it because it's crippled by the virus?
Maybe I don't know of any specific CD8 T cell specific inhibition. I'm trying to think if there was any class 1 inhibitor. And I'm not recalling any off the top of my head. Most of the inhibitors that I am aware of are inhibiting innate immune responses. But of course, if you inhibit those innate immune responses, that is going to mess up your adaptive.
There was a recent paper from Mark Davis's group out of Stanford, does a lot of these human immunology studies. And it was quite controversial. There's a lot of people that made, I don't know if you guys saw this, but made thread tweets about it, suggesting that somebody previously infected with SARS-CoV-2, they had a decreased T cell response to vaccination than those who were not previously infected with SARS-CoV-2. Yeah. I think there was a lot to learn from that paper. I don't know if those conclusions necessarily we should, go around saying to, stating that's the truth. But so I don't know. I didn't read the paper yet, so that's why I'm not fully commenting.
So
there's some literature out there.
So how's this for a question? This kind of T cell is not detected in 30% of COVID cases. Is that a good question?
I think it's a good question.
Only you and I would get it on the...
I was going to say, if people haven't listened to this episode of Immune or read this...
Yes, right.
read this review, they might not get it, but I think it's a great question.
I'm always thinking of questions when I'm reading.
What is this? Oh, for class, you're saying?
No, we're thinking about a TWIV Jeopardy.
TWIV 1000.
Yeah. Oh, yes. Oh. You guys are getting together.
But we're supposed to surprise the audience, but nobody will listen. Oh. That's it.
To our podcast? Yes, they will.
Well, when I was on Jeopardy, they asked me a COVID question. So
I know that was a pretty wild.
Okay. So this is interesting. People who are hospitalized, multiple studies of people hospitalized with COVID, they have low SARS-CoV-2 specific CD8 responses. And they say maybe this means that a weak response predisposes you to more severe disease, right?
Right.
And so that it's important for the control of infection. There are other explanations, right? But that's an interesting one.
Be nice to know, what is the adjuvant we need to boost the CD8 T cell responses? I don't know.
Yeah.
But it's also interesting to think about how that relates to levels of virus. Because in other settings, as they mention, CD8 T cell levels correlate with the amount of virus, as if the CD8 T cells are being increased by a higher viral load.
Yeah.
They sort of contrast that here, that it doesn't seem as though the high viral load is driving a high level of CD8 T cells. And that's really weird.
It's weird.
And it's different from antibodies and B cells, right? Because it seems in severe patients, there's a correlation. They have higher levels.
Yeah.
Okay. Yeah.
So each person This is pretty cool. Their CD8 T cells recognize a median of 17 different epitopes.
I was going to ask what you guys thought about that because I'm like, what do I need to know? I mean, it's good to know, but like, if
well,
it was like
it's
15,
not one or two. It's not thousands. I think that gives you a kind of a ballpark, right?
To say.
That's
yeah. Well, and I think it's important. I like to use numbers like this when talking about this because I like to tell people about why I do or don't worry about variants. And so I might talk about the fact that, if you have a mutation, maybe you get rid of one of those responses. So now you have one fewer of your 16. It's not as if you've gotten rid of all of the responses.
Yeah. I understand. Okay. Mm-hmm.
And the immunodominant proteins are spike, nucleocapsid, and M, structural proteins. And as they say, these epitopes are, in general, well conserved between variants, as Brianne just said. Yeah. They also make this interesting statement. Frequencies of circulating memory CD8 cells after infection are lower than we see for influenza. However, most of the flu studies have been done and people who've been exposed many, many times throughout their lives, and that could make a difference. But this is a cool section here. The frequency of CD8s that you need to control an infection depends on how rapid clinical disease progresses, right?
Right.
And they point out that progression of hospitalization with COVID is pretty slow. It's like 10 days post-infection or later. And so you might not need a lot of circulating CD8 cells at the time of infection, right? These could amplify in those 10 days or so, and it might be sufficient.
And I like the statement they say here that, for the memory CD8 T cells, within 72 hours, they can increase in number close to 1,000-fold.
Yeah, that's cool.
And I thought that was pretty cool. And that really hammers home for me why having a vaccine on board is a good idea beforehand, before getting actually infected. We were saying that from the beginning of the pandemic.
Yeah, sure.
Get the vaccine first so that when you're exposed, you're going to be better protected.
Exactly.
And I think that is illustrative of that. These memory T cells are there somewhere, and they can be boosted and increased by 1,000-fold in just a couple of days. And so they can pretty rapidly control the infection.
Right. Which really is, the point of vaccines is without them, you're spinning a wheel of
a lot longer,
chance, depending on all the things, comorbidities, age, and you hope you're not in the proportion that would
absolutely
be affected,
it's a roulette, right?
Yeah.
Russian roulette.
Yeah.
They also point out that it isn't fair to expect CD8s are going to reduce transmission because most of it occurs within the first 5 days, right?
Right.
And they say it's too high a bar to do that, and probably requires a large number of tissue-resident CD8 cells in the upper tract.
Which I learned from this review that if I wanted to get into an area with very little evidence, I should start trying to get nasal samples or oral samples because there's not that much known in the upper respiratory tract for these cell types.
No, not at all.
It's very important because that's where it starts, right?
Right.
It's very important. So these tissue-resident memory cells, the ones that stay in the tissues, as opposed to in the blood, they point out that these haven't been studied much because blood is so easy to get, right? And the other tissues are not. And so there are limited numbers of studies on nasal passages, oral cavity, throat, lungs even. There are some with lymph nodes because you can get samples from them. For example, they've found SARS-CoV-2 specific CD8s in lung lymph nodes, 4 out of 4 subjects, and in lung tissue from 3 or 4 out of 4. So small numbers, right? Because it's not easy to get. But they say blood may underreport TRM, right, in previously infected individuals. And this is a big gap, Steph, if you want to move into this. A big gap
We're working on getting some cool pediatric samples, right? Because they get their tonsils taken out.
Yep.
They have operations where we're getting some of those. So we might be able to learn a little bit about
that's one.
that.
They also point out pediatric T-cell memory. They say there's a lot of conflicting data.
Yeah.
One study says one thing, another study says another. Then we have this hyperinflammatory thing, multi-system inflammatory syndrome, and what's causing that. So also, I think that's a good thing to move into. Yeah.
Okay. I'll write it on my board back here. I plan.
They talk a little bit about long COVID. And
even less. Oh my gosh. I just wish.
Big knowledge gap. And so, they say, well, maybe there's persistence of viral RNA and protein in certain sites. They say maybe it could be the gut or it could be something else that's driving. But
I have a question for you, Vincent, on this.
Yeah.
I am just endlessly fascinated by these. So we'll stick to coronaviruses. I was at a talk for a murine coronavirus that has mostly brain tropism. And there's an acute infection where you can detect viral titers, and then it goes down to very low levels, but is persistent over time.
That's right.
And so maybe this is also happening in the gut. We see
it could be
antigen expression, but they can't find. I'm just like, what is the virus doing? What do you think? Because these are not viruses that integrate. These are not viruses that.
No, they don't.
Right.
So it's clear for many RNA viruses, and I think Diane Griffin was the first to show this, that you can get long-term persistence at very low levels, as you say, and you can find infectious virus, mostly in mouse models. So what's going on in the gut? I mean, that few studies I have seen, they have an EM with one particle, but I don't know what one particle means per cell even. It doesn't seem like enough. And people tell me, it's very hard to detect infectious virus, but that's not an excuse for not trying it. So I think if it were there in large amounts, it would be easy to find. Right. But I don't think it is in large amounts, and I'm not sure.
Like the virus has an ability to sustain itself even though it's replicating.
Well, with Ebola, it can hang out in the ocular fluid, in testes for years, apparently multiplying one or two or three times. If you look at the sequence changes, right?
So maybe there's a certain cell type in the gut, right?
That's unprecedented, right? How does a virus just hang around for so long?
Yeah, right? I mean, is the nucleic acid kind of just chilling in the cytoplasm and not getting degraded?
Right? That's why
maybe the particles are there. I mean, these immunoprivileged sites, there's nothing to kick it out, right? So, but the gut is not immunoprivileged.
The gut is odd.
So I don't know. I think there's still people who conclude that it's persistence in the gut. I think it's premature. I think we need more data.
Sure.
And they say it's unclear why CD8s would not clear the infection within a few weeks. So they say there's a big knowledge gap. And I just think we need a good model for this, right, where we can do some detailed studies, because you can only get so much getting samples from people,
right?
From gastroenterology, convenience samples and so forth.
But it is an interesting hypothesis what you were saying, that if there is long-term antigen production from these in the gut, could that stimulate long COVID sequelae? Yeah, I don't know.
I don't know. And why in certain people, right?
Yeah.
Right.
Okay, now they talk about CD8 T-cell memory to vaccination. 70 to 90% of people who get the 2-dose mRNA vaccines show spike-specific CD8 T-cell response. A spike because that's the one protein the mRNA encodes spike only.
Yep.
And 41 to 65% of people at 6 months after the second dose have memory CD8 T cells. And they say there's similar findings in a sense of CD8 T cells between mRNA vaccination and infection, although the magnitude with mRNA vaccines is lower than infection.
Which would that make so that's brought up a lot about the difference between infection and vaccination.
Yeah.
Yep. Yeah.
Infection is going to result in more proteins at higher levels in the germinal center for a longer time. So this is the immunological explanation, at least one of them, for why we would see bigger breadth, more cells. Okay.
What's interesting is that they mentioned this somewhere. Even asymptomatic infections where they think it's lower levels, which is one reason why it's asymptomatic, still gives you a good immune response despite it being asymptomatic. So I don't know what the cutoff is to get a good response. It's not, you don't have to have a huge viral load, apparently.
Although it's lower for B cells and antibodies, right? Like if we're just talking T cells, it's the same. But okay.
Okay.
I think it's cool that the vaccine is generating robust CD8 T cell responses at all, right?
Yeah.
Because typically we've thought for traditional vaccines of he-killed virus or whatever, they don't induce very good T cell responses.
Yeah.
In general.
But I think that was the idea of doing nucleic acid-based vaccines.
Correct. Right.
Was, the nucleic acid-based vaccines were supposed to get antigen made inside the cells so that antigen could be presented on class 1.
Exactly.
Right.
So I think it's great. It's a really the different type of vaccine platform that was able to generate these robust CD8 T cell responses is probably why this particular vaccine for this infection has been so successful.
Right.
So they point out that this first 6 months are the key for T cell memory, because there's when the cells go down the fastest. And they say, so we have twofold declines at 6 months after vaccination. That's actually good news. That suggests that memory could last many years.
Yep. Yeah.
They also mentioned some other vaccines, Chadox and the Ad26. Remember those?
Mm-hmm.
They do similar
Not so good.
elicitation of spike-specific responses.
But
50
significantly lower, right?
Yes, 50 to 64% as opposed to 70 to 90. Yeah, so it's substantially lower. And then it drops. Interesting, in people over 65, it drops to 24 to 36%.
Which I guess is another validation of Brianne's point that this platform is just not doing the same thing, right? The mechanism is different for delivery of the antigen.
Yeah, and if people are listening to all of this, also they should note that figure 2 shows all of this in picture form.
It's very nice.
And it's in some ways, it's really nice to sort of look at and think about it in a different way.
So in summary of all this, CD8 memory to mRNA and adenoviral vectored vaccines are similar in magnitude and percent responders, but individual studies vary. They say we need more head-to-head studies. Oh boy, we've had so many, right? The epitopes are varied as well. And these CD8 cells will still recognize Omicron, as we already said. The epitope recognition doesn't change. All right, so what about hybrid immunity? So we talked about infection, we've talked about vaccination, just to keep you organized there. What about hybrid immunity? Modest differences have been observed between vaccination alone and hybrid immunity for circulating spike-specific CD8 T cells. Circulating. Of course, I don't know about the tissue resident, right? Would we expect that to be different? Probably not, because this is just a vaccine, right?
Yeah. I don't know.
Or it would be, I don't, that is tough, because we would assume that in an infection, they may not be concordant. You may have higher levels of these cells in the tissue. But for vaccination, I don't know if they're going there, Yeah, there is some leakage of the antibodies, which we'll talk about in mucosal sites, but I don't know about the cells, if they're
I don't know.
getting the signals to traffic there.
Yeah, so it's hard to know whether vaccination versus infection versus hybrid would be at all comparable in those resident cells.
Who wants to donate their lung tissue?
I guess in the muscle, you got the injection, you might find some tissue resident memory cells, right? Because it was injected right in there.
There are the signals. So to upregulate these chemokines and cytokines to traffic to tissues, they do need the cytokine secretions from that mucosal tissue that muscle just doesn't secrete. So there may be cells there, you're right, but they're not going to go to the lung. Yeah.
All right, on to CD4s. How are we doing here? We are 45 minutes because we have to do CD4 and then there's some other interesting things.
Memory and antibodies.
So a lot of different kinds of CD4 cells with different functions, which Many of you have heard TH1, TH2, TH17, TFH. I've come to like TFH a lot.
Mm-hmm.
They're really cool.
Very important.
They're very cool. When I was a student, never heard of them.
Yep.
So they're all very important. So what do we have here? Infection. Oh, this is cool. Memory CD4 T cells are detectable in 93% of COVID-19 cases 1 month after infection, and still 92% at 6 months. Durable. Half-life is estimated 94 to 207 days for these CD4 T cells. And they say with the T half-life likely increasing over time. So those cells continue to develop, don't they?
Well, all of the short-lived ones died off first, so the only ones that are left are the long-lived ones.
Yep.
Mm-hmm.
And they say all these data are consistent with T cell memory being detected to SARS-1 at 17 years post-infection. These cells that are detected after infection, mostly TH1, TFH, and this is one I didn't know about, cytotoxic CD4 cells.
Yep.
Tell me about CD4 CTLs. What the hell? Why are they moving out of their lane?
They express granzyme,
yeah,
and they're capable of killing target cells that present antigens on MHC class 2. I think for me, the surprising part was just a little bit later. They say, they're of interest because SARS-CoV-2 infected epithelial cells upregulate class 2.
Yeah.
And I thought that was pretty interesting because generally we think of MHC class 2 is restricted to professional antigen presenting cells and B cells and dendritic cells, macrophages. And that MHC class 1 is pretty much on every cell. And that distinction is made because we don't want just any cell being able to present antigen to activate B cells. We'd like that to be restricted to innate immune responses through those antigen-presenting cells, so that that's the discriminator to know that something is foreign or dangerous, right? And it just an epithelial cell wouldn't necessarily be able to make that distinction.
So
although that's not an absolute, but apparently they can upregulate MHC class 2 and have this ability to activate these cytolytic CD4 T cells.
Maybe it's an extra measure of protection there at the barrier, right?
Yeah. Maybe.
So they're recognizing antigen in by T cell receptor, right?
Yeah, as far as I know.
Because they say maybe these people who have low CD8 responses, maybe they're depending on CD4 CTLs instead. That's an interesting idea, right?
That is interesting. So it's odd. So there's the same antigens being presented, obviously, in two different ways via MHC1 for CD8s or MHC2 for CD4s. But the comparison that you have 90% of individuals who are now positive versus maybe 50 in some cohorts or 30 in some cohorts, that's interesting, right? So what do we think is going on there?
I don't think they were all like 90% were CD4 CTLs, right?
No,
They're
sorry,
all the
but
different kinds of
positive
CD4 T cells.
Spike specific T cells.
Yeah.
I guess I'm not totally shocked, largely because, and this is total bias on my part of things I've spent a lot of time thinking about in graduate school. One of the things that CD4 cells do is make cytokines that help CD8s.
Correct.
And so if you have a lot of CD4s making those cytokines, they could help a very small number of memory CD8s make a good memory response, even if that small number is, say, undetectable.
Plus, you need them for making antibody too, right?
Exactly.
You need them for making
really
antibodies. Yeah.
Really
you need
important.
them for activating some of the innate immune cells too. So, yeah.
They're just amazing, those CD4s. I think they're underappreciated from my view. Anyway, they find these have been found in bone marrow, spleen, lung, lymph nodes for over 6 months. So that's good. But don't know much about these in the upper respiratory tract and the oral cavity. So there you go, more need for study. 19 epitopes on average per individual are recognized, mostly structural proteins. And again, they're conserved among variants. they make a point in this section talking about, the wide range of clinical outcomes of COVID, asymptomatic, severe disease, death. And you also get a lot of variation in CD4 T cell responses. They're influenced by old age. There's no difference between the sexes, but other comorbidities can affect them. Even healthy young men that they've, in one study, where the T cell responses are still widely heterogeneous. So something else is going on, and we don't understand what it is, Mr. Jones. Do we?
Where are our geneticists to help?
Yeah, true. That might be part of it, but it's a lot. So it's hard to know that. I don't know.
Mm-hmm.
Here's the part I like. Asymptomatic SARS-CoV-2 may reflect shorter infections with lower overall viral loads in tissues, which could be expected to have lower T cell memory as a result of lower antigen exposure. So maybe that's part of it in these young people getting asymptomatic infections. The heterogeneity in the T cell response may be a consequence. I mean, that's an idea. I think you'd have to get some data, right?
Yeah.
Right. It does go back. Yeah. Right. Bringing up that viral load question, and would the T cell data reflect the B cell and antibody data in terms of severity of disease? But you're right. The study you'd want to do is that you could give people different doses, and then, but we don't really want to do that. We don't really want human challenge.
There was a whole area of literature on this where they tried to look at length of time of antigen persistence and amounts of antigen with bacterial infections and treating with antibiotics at different times to see how memory was changed by the antigen persisting for different levels of amounts of time. But of course, you can't really do that in this situation.
Yeah.
I can tell you that when I had a coronavirus that infected pigs and I gave them more virus, they had a larger immune response, more cells, more antibodies, more T cells. So it's a different coronavirus in pigs, but
I think the principles are similar. So the infection proceeded with different kinetics also when you gave them more virus?
They shed higher titers in the gut. Mm-hmm.
But they didn't, the incubation period wasn't shortened or anything like that?
No, correct. Okay. Just the magnitude was higher.
These are the 600-pound pigs you used to wrestle, right?
Yeah. Do you remember that? That
was when we
started this podcast. Yeah.
I love that thought of you in there, grabbing.
And it's the reason why now I subcontract my pig workout to
you don't want to
veterinary
do that
school, so I don't have to do that.
No, you had the experience. That's enough.
Yeah.
There's a whole section here on cross-reactive memory T cells. And they found, so it's been found that T cells, CD4s that recognize SARS-CoV-2 can be found in people, blood from people who were taken before the pandemic.
Didn't we talk about some of this? Did you do a paper on this, Brianne? I sort of remember us talking about this. And we had some issues with what they were defining as pre-existing memory, if I remember that discussion.
I sort of remember that as well. Yeah. I think that they just decided that detectable meant that it was memory, if I remember correctly.
I think that was
it had to have been amplified enough to be detectable, so therefore it must be memory.
Yeah, so I guess I didn't go look at the papers that they reference here. There seem to be quite a few, so maybe there's additional things that have been done since then to see.
I mean, these seem to be these recognized SARS-CoV-2 proteins, of course, but also the common cold coronaviruses. So that's the idea that they originated from that. And they say, which is the right question, we don't know if this is biologically relevant. Does this make a difference? And they say, well, there are some studies that they may, having pre-existing cross-reactive T cells may make a difference. For example, in one study, the presence of these were linked to favorable outcomes in a large healthcare worker cohort. It's an association, so you don't know if that's the only thing that made a difference. So they say there's some suggestion that they may make a difference, but I don't know if it's convincing. What do you guys think?
I think it's really hard to make conclusions about this because you have to have some people who have never been exposed to those common cold coronaviruses.
Yes, that's right.
That would be hard to do.
We don't really have a cohort that clearly has not been exposed so that we can make those comparisons.
Yeah. So if you had an animal model where you could infect or not with a common cold corona, you get immunity, then you could challenge that would tell you in principle that you could get cross protection, but we don't have such models. Yeah.
Right.
No.
Do we know?
Not
Do
that I've
the
seen.
common colds infect monkeys? I don't know if I've seen a.
No idea.
I don't know.
Okay.
I don't know. I don't know if
That
anyone.
would be the animal I would try if I had unlimited resources.
So expensive.
Yeah.
It's so cute.
But there are, at least in terms of CD8 responses, there are cases of viruses that you wouldn't really expect to cross-react in terms of they're in different viral families. They're very different. But if you actually do the bioinformatics, you find that they happen to have an epitope or even certain amino acids that are similar, and you get cross-reactivity and influences on memory responses. So I know that Pekindi and CMV, for example, will show some of those. So you could also at least do some bioinformatics to get some ideas of where you might see some of those cross-reactive responses before you went into the animal studies.
What was the first thing you said? Pekindi?
Yeah.
I think that's how you pronounce it.
Is it a virus?
It is a virus.
Oh, okay.
I used to call it Pichinde, but it's fine. Pichinde. Yeah.
Yeah, I largely know about it from those papers.
Right. Okay. Interesting.
So.
All right, let me try and move a little quicker. CD4 T cell, that's not, you're fine, to vaccination. Let me summarize this. The vaccines elicit robust CD4 T cell memory. Reductions over 6 months are modest. They say overall, each major subset is maintained for at least 6 months, subset of CD4 T cells after vaccination with the 2 mRNA vaccines and the Ad26 and with the Novavax protein-based vaccine. And they say with kinetics that indicate they should be maintained for years. So that's good. We don't have much data for other vaccines like Coronavac, Sinopharm, and Sputnik. And again, the epitopes are conserved in all the variants. All right, now hybrid immunity. CD4 T cell memory in hybrid immunity. Modest differences between vaccination only and hybrid immunity for circulating spike-specific memory CD4 T cells. No difference based on symptomatic or asymptomatic infection. There is a dramatic enhancement of antibody B cell responses in these people, which we're going to come to in a bit. How about the location of the memory? As we were just talking about, intramuscular vaccination, they say, is expected to give you mostly circulating T cell memory, whereas infection gives you both circulating and tissue resident, right? So they say, okay, so the hybrid immunity should give you both, but we don't know if the vaccines actually enhance tissue resident memory that are already there from infection. And if the order is vaccine and then infection, it's not known whether the TRMs are different in any way that would happen after infection alone.
Right.
All right, B cells. They can be very long-lived. They say smallpox B memory cells can last over 50 years, and those against 1918 influenza at least 90 years.
It's very cool. It's very cool. Look at our immune systems.
And they can react within 3 to 5 days and start making antibodies. And I love this sentence here. So the first importance, they say, is to make antibodies, which are going to block infection, to make a memory response. But the second is to serve as a library of predictions by the immune system of possible future variants. And so as these B cells are dividing, that's what they're doing. And they say it illustrates the brilliance of the immune system at predicting viral mutations. The COVID-19 pandemic has illustrated the importance of B cell memory in recognizing all of these variants. So I thought that was cool. The brilliance of the immune system. You're not going to argue with that, right?
No. It's had a lot of millions of years to figure it out.
Yep.
I was thinking, if it weren't brilliant, we'd be dead, right? It has to be really good because there's huge pressure. I mean, look at all the pathogens are just microbes around us, viruses, bacteria, fungi, constantly assaulting us, right?
and making new generations with new genetic combinations a whole lot faster than we're making new generations.
Yes, yes.
So here's infection, B-cell memory and infection. Detectable B memory cells you develop within 2 weeks of symptom onset. That's very interesting. So not infection, but symptom onset. And the frequencies of memory B cells can increase continuously over 3 to 6 months post-infection. And these increases are associated with somatic hypermutation that goes on for at least 12 months. Increased affinity, increased neutralization and potency, particularly against variants. And they say these are all indicative of long-lasting germinal centers after infection, except remember that paper we did. Did we do it on TWIV, Brianne?
I think we did it on TWIV.
Fatal COVID, where you have disruption of germinal centers, right?
Yeah.
In autopsies.
Mm-hmm.
This is also interesting. IgM cells come up initially. They're 1/3 of the B memory. They decline very quickly. That you don't find them after 6 months. IgA positive B memory cells are uncommon. Only 5% of spike or RBD specific B memory cells on the average, but they are maintained in the blood. Yeah. So you're going to look.
I think they're probably more going
in
on
the mucosa. Yeah. So in the mucosal sites, you think they're memory IgA cells? Yeah.
Yeah, I would. That would be my hypothesis.
But even though they're few, they're maintained for 8 months, which is good. Severity of disease impacts the magnitude of the response. If you're hospitalized with COVID, you develop higher B memory cells against the receptor binding domain compared to people with mild COVID, which is the same as you would see with antibody titers. But asymptomatic people develop similar spike-specific memory cell frequencies compared to symptomatic but non-hospitalized. So overall, the memory response they say, is quite impressive. Substantial receptor binding domain and spike-specific B memory cell generation, and with exposure just to one strain, the B-cell memory compartment develops over months to contain cells with high neutralization potency and cells capable of recognizing and neutralizing a range of variants. They did not have, a knowledge gap is upper respiratory tissues, where they don't know about B memory cells, as we've just said, because that's the first place the virus goes. So we should look. There you go, Steph.
Okay.
And there's no SARS-CoV-2 specific B memory study reported for long COVID patients.
Although I know people are working on it, so hopefully we'd have something.
All right, so that was infection. Now vaccination. This is quite interesting. 2-dose mRNA vaccines give you what they call substantial affinity matured B memory cells, yet it is still qualitatively poorer than after infection. You get improvements in neutralizing antibody breath months after infection, but not after RNA vaccination. And listen to this. These differences may be related to the narrow time between dose 1 and dose 2 of the RNA vaccines.
Right. You need that antigen to be in the germinal center for a longer period of time, which is what the infection does.
They say that if you interrupt it, you're interrupting the maturation, right? By giving a second dose. You're distracting all those cells. Someone else is coming in.
So that's the sad part about you don't have time in a pandemic to know that. But I wonder, in the next pandemic, would we space those apart longer?
We know, right?
I think it's hard to tell because while yes, you would get a certainly quantitatively better response by spacing them better, you still would be leaving people with sort of the reduced protection of one dose.
I do see
for a longer period of
time.
So you're right.
During the pandemic. Like in that time when you're trying to protect people because hospitals are overrun, the virus is circulating. I see what you're saying.
Yeah. Are you trying to go for protection now during the height of the pandemic, or responses that are going to give you the best memory in 5 years?
That's a good point.
There have been modeling studies that address that, because they're talking about going from 3 weeks to 10 weeks between the doses. That would substantially get higher titers, and also, the higher titers are what's important, because the variants didn't come out until the end of that first year. So initially you're not worried about the variants. It's just higher antibody titers, right? They also have some papers on Ad26 vaccine and Novavax. They both give you lower B-cell memory frequencies than the mRNA vaccines.
I feel bad for those people that work for that company. It's like, not as good.
But what they don't ever say is it better than the other types of vaccines that we've been using in the past?
Yeah.
Right? Because we're now comparing it to this mRNA vaccine that seems to be the superstar.
Sure.
Yeah, but I think we'd have to have some of those other vaccine platforms for this antigen.
There are some, there's like one paragraph on protein recombinant protein vaccines that they actually do a pretty good job, if I'm remembering that correctly.
Yeah, it's not terrible.
But anyhow.
For antibodies.
We'll get there.
Right. For antibodies.
Right.
Of course, germinal centers are important for these responses, and they say these are reduced in many immunocompromised individuals, like kidney transplant recipients. There's a whole section on that later. All right. So that was vaccination. Now, B-cell memory and hybrid immunity. This is increased. The circulating spike and RBD, IgG, B memory cells increase substantially in hybrid immunity, but become similar to 2-dose vaccination after 6 months. So in hybrid immunity, the B memory cells have more somatic hypermutation and affinity maturation than after vaccination alone. And this can occur after infection plus vaccination, or vaccination plus infection, which we're going to talk about in a bit. Neutralizing antibody titers stabilize between 4 and 6 months with little evidence of decline thereafter. And
so we think that those long-lived plasma cells have like some set point, and then they get there and they're like, all right.
The half-life is 254 days. It's pretty good. Neutralizing antibody titers are detectable in approximately 80 to 90% of SARS-CoV-2 infected people at 6 and 12 months post-infection. If previously infected only, the titers are relatively low. So they're boosted by this mixture. Okay. Long-term antibody titers are lower in asymptomatic cases at 6 to 16 months. That's kind of interesting. Some people are actually seronegative, although they say some of this may be due to false positive PCR tests with high CT values.
Interesting.
Oh, here's a good one. Nucleocapsid antibody assays are not trustworthy indicators of previous infection over 6 months after infection. If you're using them over 6 months.
That's interesting.
They say this may be because the antibodies are decaying quicker, or maybe cross-reactive antibodies are interferon, but they say you should use IgG against the receptor binding domain, although that won't distinguish infection versus vaccination.
Well, I think it's fascinating that we can actually make comments like this paragraph and one before, where we can really get to look how much we get a different answer when we use different techniques. And, we can really robustly say that with this much data.
Yeah. And so we are assuming that nucleocapsid antibodies in an infection are quantitatively lower than spike, because less of that protein is produced than spike. Or do we not know that? I can't assume that.
Sorry, say that again.
So are nucleocapsid antibodies produced in lower quantities than spike in an infection?
I don't know that. That's a good question.
I just wonder.
I think so.
I would assume so. In my mind, I'm thinking spikes produce the most, so that would make sense. But I'm just thinking if they are degraded at different rates, that's fascinating. And that would suggest that they're different.
Nucleocapsid is the most abundant protein. in an infected cell.
Yeah.
Okay, so then it's maybe the reason. So then we can't assume that they're at lower quantities than spike.
No.
Okay. Because I was just wondering if they are, maybe it's right, a detection issue. They get so low that the assays can't detect them anymore. But if it is really a difference in degradation that suggests they have different glycosylation on the FC portion, which is is fascinating.
Yeah. I mean, and in other situations, those types of hierarchies involve things like binding strength and other cytokines that the cells get when they get activated. So I could imagine those degradation rates could differ based on those same kinds of principles.
Okay.
Like the signal strength that the B cell got when it
from that
was activated.
Antigen.
Yeah, from that antigen, whether it's because of other cytokines or whether it's because of the actual antibody antigen binding.
Right. Okay. Yep. Gotcha.
They say local immunity is important, right? So your mucosal tissues, if you have antibodies there, that's going to have an impact on infection. And they say circulating IgG can move from the circulation into mucosal surfaces and provide protection. There are local production of IgA and IgG in salivary tissues for a limited number of months post-infection, but not really has been studied all that much. So there you go, Steph. That's what Steph is going to do.
I'll get on that.
All right. Well, antibody durability. How long do antibodies last after vaccination? They said, so as we said, 2 doses are really good at making high titers of neutralizing antibodies, but the biggest shortcoming is that they decline over a period of months. And but you can find plasma cells specific for the spike after 6 months in 2-dose mRNA vaccinated people. And they say, but still the antibody titers are declining. So maybe these are low frequency plasma cells, or they don't quite understand that. And so they say because of this drop in antibody titers and the emergence of variants, we have gone to 3-dose mRNA vaccine regimens. And the question is, do they make more durable antibody responses than 2-dose regimens, right? They have a wonderful discussion here, which says we can make durable antibody responses. The infected people do that, right? So they say that, and many vaccines are 3-dose regimens. They say one can consider that this is because the immune system performs a cost-benefit analysis of durable memory to each antigen exposure. Durable antibody responses for 10 years or more have a high caloric resource cost commitment, whereas durable B memory cells or T cells have lower caloric costs. As such, it frequently takes multiple antigen exposures to trigger significant durable antibody responses. So I think that's pretty cool that you think of it in that sense, right?
Yeah, that's very interesting. Right. So basically what we're saying is that it's too costly if you're not going to see this virus again to make a long-lived plasma cell, to take all the time in the germinal center. Mm-hmm. So seeing it a couple times.
And to give that plasma cell all the resources it will need to survive.
Yeah.
And to keep pumping out antibodies.
Good way of looking at it. Anyway, they say the studies are conflicting. The conclusions about durability of antibodies after 3 doses remain uncertain. Mm-hmm. So we just don't know.
Just a quick question for those who are reading this. When they say acute antibody responses, and then they just start saying AC, but they don't define. Does that mean acute? And they just decided to chop off the UTE?
I think so.
In that paragraph?
Yes.
Okay.
I think so.
Because there's no effort to try to tell me that. I just was
I noticed, there's a lot of
yeah, but it's fine.
Yeah.
And by the way, the adenovectored vaccines give you lower antibody responses compared with the mRNA vaccines. And the protein-based vaccines don't do badly. Actually.
Yeah, that was interesting. We have very little data, but they said neutralizing antibody titers 6 months after the second dose are equivalent between one of the protein-based vaccines and then the 2 mRNAs.
Yeah. And they say mixing and matching could actually be good, but we don't have enough data. So peak neutralizing antibodies you get after 2-dose mRNA or protein vaccines. And the adenovectors give you more stable responses at 6 months. So maybe mixing the two would give you the best of both, but they haven't been studied all that much. And then finally, local antibodies, again, in the respiratory tract. There's evidence that the mRNA vaccines elicit some circulating IgA. You can find IgA and G in saliva and nasal swabs, but they decline quickly over 6 months after 2 doses of mRNA. So they say exploring development of intranasal vaccines is of substantial interest for improving mucosal immune memory. I think that's quite clear, right?
Yeah. When we get to the end, I can tell a little story about my experience of watching companies trying to get mucosal vaccines to market. It's been interesting to watch. I can give a little anecdote.
All right, just a couple more sections here. We have antibody durability and hybrid immunity. Let's see. All right, if I could summarize this. The most prominent characteristic of hybrid immunity is the impressive improvement in neutralizing titers and the breadth of neutralization. We've already said that as well. How about durability? Where's the durability? Antibody durability at 6 months is robust in a majority of individuals with hybrid immunity. After 6 months, hybrid immunity, the antibodies maintained 5 to 17-fold higher compared to people who were 2-dose mRNA vaccinated or infected alone. And you get higher nasal IgG and IgA in these people with hybrid immunity, either infected or vaccinated plus infected at 10 months. So hybrid immunity is really good for durability too.
And eventually everybody's going to have that, right?
Yeah, for sure.
It seems to be finding everyone.
For sure.
Especially since the beginning of the year, I think.
All right, 2 more sections. One is interesting, immune memory and immunocompromised or immunosuppressed people, right? So they say this is very important. It varies depending on the specific situation. So you have some people who are depleted for B cells, like they're getting anti-CD20 monoclonal treatment. They have defective antibody, B memory, and T follicular helper cell responses, but their Th1 and CD8 T cell responses are normal or even elevated. So we expect that they're going to have defects in immune memory. Solid organ transplant patients, they're immunosuppressed. We don't know a lot about them, but there seems to be severe memory defects in them. And some cancer patients also are on immunosuppressive regimens, in particular fingolimod causes an almost complete block of antibody and T cell responses to COVID vaccines. It's not good.
No.
Which makes sense that is impacting NFAT, a transcription factor that's important for both B cells and T cells.
The fingolimod, that's a S1P receptor antagonist.
Yeah.
Yeah, I looked it up. So it's a drug called Gilenya, treatment of multiple sclerosis, because basically it blocks T cell trafficking. It's frequently used in mouse models to trap T cells in the lymph node and not let them exit. But in fact, more recent studies kind of show it just parks T cells where they are and they can't get up and move around. So it pretty much blocks their access to the lymph node or egress from the lymph node. So it blocks T cell responses.
Yeah. Sorry, I got confused about that.
No, that's okay.
Whatever you said, that was interesting too. I mean,
yeah. I mean, that would block T cell responses too, blocking NFAT, because that's critical for T cell receptor signaling. So, yeah.
That is a drug I teach about on the same slide as fingolimod, I'm pretty sure.
Yeah.
So this is the last section, actually. If you haven't listened at all, and now you're going to listen, this is important. This is probably the take-home message of the whole thing, all right? Boosters in persons with hybrid immunity. I got hybrid immunity. I have 3 vaccines, and I got COVID. So this is me. I didn't take a booster, and the reason is embodied here. So hybrid immunity, as we've said, is the most robust. That includes vaccine, infected, vaccine, or infected, vaccine. I have vaccine infected.
Yeah, me
They have
too.
the best neutralizing antibody breath. They can recognize all the variants. They have better local immunity in the nose and the mouth, as we have said. And that doesn't happen by intramuscular vaccination. It only happens with this hybrid.
There's some antibodies that make it, but it's
not
less.
a lot, right? And they're more durable. They last longer. And they say, but some governments have big booster vaccination requirements within 90 days of an infection. And they said, that's too soon. And it may be counterproductive. Maybe these people don't need a booster for years, right? Because you may be disrupting the somatic hypermutation that's going on after that infection by giving a booster within 90 days. Too close together. They call it short-sighted and could result in poor quality immunity. We know that memory B cell frequencies increase for almost 6 months after infection. Germinal centers can persist for more than 6 months after infection. They can be productive for more than 6 months after vaccination. They continue to improve the quality of neutralizing antibodies for 3 to 6 months. And they end by saying boosters too close together may disrupt those processes of generating broader protection against future variants. So that's the answer to people who say, people say, I'm going to Florida. Should I get my third booster?
It depends. Were they infected?
Yeah, if they were
Do they have
vaccinated.
compromised?
Are they older?
Well,
I'm hesitant to say no boosters for anyone because there are people.
Yeah, but most people are vaccinated at least 3 times. So if you've been vaccinated 3 times and never infected, I don't know what the answer is because I don't know. I'm not sure I saw a study in here that looks at that.
Well, you were saying the booster is the third dose. So if somebody's asking me if they should get a booster or third dose and they are older or whatever, sure. After that, I don't have a...
Definitely
I'm talking about
have
the
it 3
bivalent.
doses.
Yeah, I'm
so
talking
you're
about
talking
the bivalent. Yeah, so the third booster is the one that was, that corrected the first 2 being too close. Right.
So you're not talking about that because that I would...
Oh yeah, that's for sure. I totally agree.
People.
And we got that.
I had that.
That was the original, the original virus we got.
But you mean the bivalent updated booster? I got
yeah. So if you had, say, 2 vaccines, a booster, and then got infected,
yeah, you're fine.
I don't think. Although Daniel Griffin would say, if you're over 80 or 85, then you maybe should get it. But then, how long is it going to last, right?
Well, or if you're a kidney transplant recipient or something like that.
Yeah.
But you're right. You have to find out what they're. But most of the people who ask us,
right,
have had multiple vaccinations. Should I get a 5th bivalent booster? Yeah, I'm not sure. But we need to study to see if it's making any difference or if it's hurting. Right. I think this discussion is really good in terms of it interrupting the processes. Right.
For sure.
Because in the
More
real
is
world,
not necessarily better, right?
Yeah, it's true. Anyway, there was a lot there, and hopefully you got something out of it. I learned a lot. I thought it was really good.
Yeah, that's great.
I did.
Has anyone looked at the other articles in this issue?
I didn't.
It kept telling me to read Goldblatt, right?
Yep.
Yeah,
Yep.
it did.
I did not read that. Sorry to report.
Yeah, no, I did not have
but maybe I will.
I had found a couple of them sounded interesting and hoped to read them later.
Yeah.
That's number 24. Oh, yeah, Sean is on it. That's correlates of protection against SARS-CoV-2 infection and COVID-19 disease. Wow.
Mm-hmm. Wow.
I'd like to learn what are the correlates of protection. Okay.
probably CD8 T cells, CD4
Antibodies.
T cells, and antibodies.
Maybe CD4 T cells since they're so, I mean, if they're in so many people.
Yeah.
Anyway.
So do you want, I could share my mucosal.
Yeah, share your story.
Yeah,
Go ahead.
share your story.
So it's been really fascinating. I've been collaborating with this company that has a platform that's a pill-based vaccine. And you would take it, it's enterically coated, it would uncoat in your stomach and stimulate because those gut cells are going to go to other places. And there's a lot of companies doing mucosal vaccines, mostly focused on the upper respiratory tract. But it's very interesting that even though the science, the rationale is there, maybe you would want to boost, especially in older individuals. If you don't have the financial incentive, you cannot get investors to invest in the process that takes you through FDA. So you're pretty much too late at this point, because we're no longer worried about the virus circulating and causing clinically relevant disease at high levels like we were. And it is hard to demonstrate superiority over what's currently available, which is another very big component in getting something to market. So these mucosal vaccines have to demonstrate that they are better than the injectable mRNA vaccines at things like protecting against severe disease, which is a high bar. Things that we would think mucosal vaccines would be better at are transmission, but that is difficult to assess and take a very long time and money. So again, it goes back to they don't have the investors, because the investors are like, the landscape to me doesn't look like I'm going to make money if your mucosal vaccine goes to market. Yeah. And so really these mucosal vaccines, what we're learning now will be great for the next pandemic, but they need to be kind of like the mRNA vaccine, ready to go at the beginning when you have enough people to demonstrate these type of correlates. So I've just been fascinated to learn about like the industry side of things and what it actually takes to get something through.
Don't you think that having our health depend on for-profit industries is just a bad plan to begin with?
Yeah. It doesn't have the foresight to, produce things that would be helpful in the future. I mean, I don't mean to say that they're not doing that. I'm sure, like for different things, but
if they make money, they do it. It's fine. Right. But for example, we're not seeing monoclonals against any of the Omicron subvariants, because by the time they get out, there'll be a new one and they won't make any money. So that's the end of the monoclonal therapy, right? Right. I just think that I know it's worked really well, but a lot of things, diseases in other countries that are substantial for them, but they can't do it. We're not going to make it because we're not going to make it. It's just not right.
Yeah, it is very obvious that the money is what drives things that save people's lives, which is sad. And so then you have to find, okay, how do we get out of that? But we are very locked into this system. It would be very hard to.
I thought that, these nonprofits, CEPI and Ready were a good idea, but there's only two, and that's it. I thought that they would spawn others where they would raise money from donors and invest in development through a phase 1, which would be good. So CEPI, for example, has brought a Nipah vaccine through phase 1. So if there ever is a big outbreak, it'll be ready. And the only reason, if there were an outbreak, say in the U.S., we would have it. The only reason we would is because people in other countries are dying of Nipah, and CEPI thought they should have some protection, right? Right. But then in the end, we benefit, and we didn't even make much of an investment. So I don't know how you change that. I think it's really hard. But I feel really badly that people in wealthy countries get to be healthy and other people do not, because we're all humans and we all deserve to have good lives based on the benefits that our brains have given all of us, right? Right.
So what do we think so the solution could be? Really, it's like you need very I'm trying to think of the word. Ethical, but like altruistic. Thank you. People who have a lot of money who can do this. And there are some, like the Bill and Melinda Gates Foundation, they fund diarrheal diseases in third world countries that wouldn't normally get funding. So that's that's one of them, right?
They fund the polio eradication effort, and Warren Buffett does something. But there are a lot of billionaires who don't do anything. And they're the people who have more money in the world than anyone else. They, in my opinion, they should be helping others, but they just buy bigger boats and bigger cars,
or they invest in things that are going to make
more money.
Right. Like the goal is to make more money. So it is challenging to kind of try to extract people who have made billions because they're in that system, right? The system, to try to pull them out.
Well, and we can also see sort of how well kind of the approach of having the VRC has worked out in terms of thinking about the mRNA vaccines and things like that.
Can you explain that to people maybe who don't know what that is?
Sure.
I know, but for that.
So the VRC, or the Vaccine Research Center, is an NIH-funded group of scientists that are working on a lot of vaccine-related issues, vaccine platforms, things like that. And that is where the mRNA vaccine that partnered with Moderna was originated. And it was largely through thinking about really basic science types of questions in vaccinology. It was the VRC originated to deal with HIV, but many interesting basic science questions about vaccinology, questions about MERS vaccines or Nipah vaccines have been described. But that meant that a lot of the technology was ready at the beginning of this pandemic. And I know that on TWIV, we talked to both Kizzmekia Corbett and John Mascola at different times, if people want to hear more about that. But so we know that if we have this group of people doing this kind of stuff, we can make good progress. Now we need to find those billionaires to fund more VRCs.
Right.
Yeah, just a plug for Microbe TV. This is
there you go.
We're trying to do this with knowledge. We're trying to give it to everybody, right? Not charge. We want people with money to donate. So far, it's just our listeners who donate, grassroots stuff. We can't get the wealthy people to be interested, but that's...
Who do I got to talk to? Who do you know? Do you know people?
I don't. I'm just a little guy with, with not a lot of connections, but I want to try and attract people to understand that the more we spread knowledge, the better off we are, because we have a big problem with truth in this society, and knowledge can counter that. So, but if you're a billionaire and you want to donate to a vaccine company, do that. That's fine. You don't have to donate to us, but we need much less money than a vaccine company.
Maybe donate to the VRC though, because the vaccine company is going to just take those profits and.
No, I'm thinking of like CEPI or Ready.
Oh, like CEPI,
right?
yes.
CEPI has done good. I mean, they funded a lot of COVID vaccine trials as well. So they moved out of just the neglected tropical diseases for a bit. But and we did a podcast with the CEO of CEPI
That's cool.
back at the beginning in 2020. And that's before the pandemic. It was actually recorded in 2019. So
oh wow,
the conversation's interesting. Whoa, we want to be ready for the next big one. Little did we know. So we were sitting there in Singapore recording it, and the virus is circulating not too far away already.
That's wild. That's wild.
In December. And I went and gave a talk at the university, and there were some students there who said, "Oh, next year we're going to have a virology meeting in Wuhan. We're going to invite you to come and do a TWIV."
Oh, how wild.
So much for that. We'll never go there now. They will never let us go do a TWIV there. What a mess. Oh, well. Okay, that is Immune 66. You can send your questions and comments to immune@microbe.tv. You can support us if you are interested in educating people for free about life sciences anyway. I can't do other things, but life sciences, go to microbe.tv/contribute. Number of ways you can do that. If everyone who listened gave $1 a month, boy, we could do so many things. Not even that much money, right? Microbe.tv/contribute. Cindy Leifer is at Cornell University. Cindy Leifer on Twitter. Thanks, Cindy.
Thank you.
Steph Langle's at Case Western Reserve University. Stephanie Langle on Twitter. Thanks, Steph.
Thanks. Yep. This is a great time.
Brianne Barker's at Drew University. BioProf Barker on Twitter. Thanks, Brianne.
Thanks. I learned a lot.
I'm Vincent Racaniello. You can find me at virology.ws. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.